welcome to Bryce Canyon 2025 Geology Festival, or GeoFest, whatever it is. Have you guys been having a good time so far? Yeah. Nice. Did anybody come here specifically for GeoFest? Nice. Okay, awesome. Is anybody here at Bryce Canyon for the very first time? Nice, cool. That's a couple of you. Did you guys have what they call the Bryce moment today? Do you know what that is? People here who work here talk about it. It's when you go out and you see the amphitheater for the first time and you're like kind of just like in awe of how amazing and huge it is and all of the formations. Did you kind of have that? A little bit? I remember my Bryce moment. I came out here for the first time a few years back with one of my geologist friends, and she and I got to Sunset Point right at sunset, and I took this photo in that exact moment. And, you know, as a geologist myself, you'd think that I would have some sort of, insightful thoughts about, the composition of the rocks or the formation of the hoodoos as my first impression of Bryce Canyon. But literally, I remember that exact moment. My first impression was, wow, it is really orange. And that was my entire first impression. So, obviously, that's why they invited me to speak here, because I'm full of very insightful geologic revelations. You know what I'm trying to say. All right. But you might be asking yourself, who even am I? Why the heck do I get to be up here on stage talking to you guys tonight? And why should you trust what I have to say? These are all very valid questions. So hello, I'm Madison. You guys can all call me Matt if you would like, because we're all friends here. And I do a lot of things. I'm a geologist. Sometimes I'm a paleontologist. And I used to be a park ranger at Badlands National Park. I also have ADHD, so I do like a lot of things. Sometimes too many things, and all the things overlap, and it's really fun. And I'm also do a lot of science communication, which is one of my favorite things to do. And it's what I'm doing right now for this presentation. And I do a lot of science communication with my partner in life, crime, and in the cowboy way, Max, who will be, guess what, tomorrow's speaker. So if you guys are still around tomorrow night, you should absolutely come back to catch his talk too, because it's going to be awesome. Let's see, why else should you trust what I have to say? Oh, the last reason, whoa, I think I just turned everything off. There we go. I'm a big nerd, which is very important when you're speaking at these things. Nikki can attest, it's true. So I could, stand up here and rattle off all my boring, stuff that I've done. I have a degree in geology. I've published a paleontology paper. I'm going to grad school, blah, 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 blah. But really, I think the main reason you should trust what I have to say is because I've been telling people since I was 3 years old that I was going to be a geologist and paleontologist. And then I did that. So I'm stubborn, and I've spent my whole life studying these things. And it's like my entire obsession. You guys can attest. Pretty much all I talk about. And since we're here at a geology festival, I hope some of you have the answer to my next question, which is, what is geology? I feel like it's pretty important we cover this. And I'm going to be asking you guys a bunch of questions. So feel free to shout stuff out as we go. I'm a big fan of audience participation in my talks. I'll give you some hints, though. Rocks, time. What else? Anybody?
Hammers.
Earth, hammers. All very good answers. Exactly. You guys get the idea. Geology is officially, whoop, I keep hitting the wrong one. There we go. The study of the earth. That includes the composition of the earth, the processes that have shaped it and continue to shape it today. Everything that pertains to the earth. So not just rocks, but rocks are a very big part of it, of course. And hammers to break the rocks. All right. Got another one for you guys since you did so well on that first question. What is biology? Anybody? Study of life. Yeah. Anyone have anything to add? No? Okay. Very good. The study of living organisms and kind of everything pertaining to them. Now, why do I ask you about biology when this is a geology festival? Well, because if you take geology and you combine it with a little bit of biology, let's say the two of them meet, they fall in love, they have a baby, that baby is paleontology. Paleontology is the perfect blend of geology and biology, with a little added math, chemistry, physics, basically any science you can think of. Paleontology has a touch of it. I like to brag that we're the most interdisciplinary science. And I know I mentioned I'm a geologist originally, but these days I mostly refer to myself as a paleontologist. So that is actually what our talk tonight is going to be mostly about. So now, with that context, what is paleontology? Anyone? Dead living things. What did you say? Old things. Paleo.
Things that are from the earth.
Old things from the earth. Yeah, biology plus geology. Exactly. So, paleontology is the study of ancient life on Earth, which is usually studied through the fossil record. So basically, life in the rocks, right? So, the perfect blend. And as I was preparing for this talking and thinking about what I wanted to talk to you guys about tonight, I kept kind of ruminating over one question. Because I'm incredibly biased, obviously. I think that paleontology is the coolest job in the world. We get to go out there, we hunt for fossils in remote parts of the country, we dig up dinosaur bones, we help name new species, all sorts of cool stuff. And then we get to go and educate about it sometimes too. It's like the best job in the world. But what I wanted to know was, what does the general public think that the coolest jobs in science are? I think it should be paleontology, but we'll see. So, like any good scientist, I decided to go about this the scientific way and take a very official poll of the general public. Decided to take a very official poll. And I decided to ask people, just a bunch of people, what they thought the answer of that question was. You got to ask a lot of people if you're going to take a poll because you want a diverse, set of backgrounds, kind of range of people so that you get the most, comprehensive and truest results. So I asked a bunch of people because you never want to assume what people are going to say. So the results are in, and I can now present to you guys this never before seen, very official data about the coolest jobs in science. So, drum roll please. Number one, obviously we've got wait. Okay, I wasn't expecting that. Astronomy beat us out. The astronauts? What? Okay, now that I'm saying that out loud, it kind of makes sense. I guess being an astronaut is pretty cool. Fine, but we better have gotten second. So, all right. Now I'm kind of nervous. Okay, second place. Drum roll, please. Oh, thank God. Okay. Whew, looks like it was pretty close, too. Oh, okay, cool. So at least we're number 2. And let's see, number 3. Oh, very nice. Archaeology. And now I think, yeah, I saw a note up here earlier. Yeah, there's an honorable mention for zoology and wildlife biology, too. So, coolest jobs in science. Would you guys agree with this poll? Can you think of anything cooler? Cryogenics. That that is cooler. But that was too good. So now that we've got our top three coolest jobs in science, even though it wasn't the exact results I wanted, we'll continue down this train of thought. Because now what I want to know is how can paleontology up our game to become the coolest science in the eyes of the general public? And to figure out how we can do that, I think we need to understand why these three are the most popular. Do you guys have any ideas about what the connecting factor between these 3 professions might be? Media. They get a lot of media coverage, but why do they get a lot of media coverage? Do you have an idea?
I think you guys just need better hats.
We need better hats? Oh, but we already have the best hats. Come on. I know, Indiana Jones has a pretty good hat. But Indiana Jones is based on a paleontologist, actually. Fun fact. Exploration. Yeah. Exploration. What else? The unknown. The thrill of discovery. That was kind of my train of thought, right? All of these really involve exploring something new, going into the unknown, answering questions that we don't have answers to yet, seeing something that nobody else has seen before. You gotta love it, right? I mean, that's what it's all about. So, I think that's why they get a lot of media coverage. So, now that we know kind of why these are the most popular sciences, and if it is the thrill of discovery and exploration, the next question I have to ask, if we're trying to make paleontology the coolest, is what is there left to discover? So I consulted the ancient texts and scrolls, and you guys aren't going to believe this, but I actually found an ancient exploration to-do list for humanity. So we'll see how we've done. Point number 1, map the entire surface of the Earth. We've done that. Boom, did it. We even have Google Earth. That's pretty crazy. You know, if we hadn't done this, I do feel like geographer or cartographer might be a job that might have made the top 3 in the past. All right, point number 2. Ooh, explore the deep ocean. Ugh, we haven't quite done that one. Even NASA tried, and they were like, literally going to space is easier, so we're gonna do that. So we don't quite have the technology to do that one yet, but maybe we'll get back to it eventually. But going to the moon, NASA did accomplish that one. In addition, we've also mapped the observable universe. We've figured out what lies beneath the Earth's crust. We know all the different layers of the Earth and the core and all of that stuff, and we've explored the poles of the planet. We've accomplished some pretty amazing things, right? So what is there left to discover? Well, it seems to me that besides the deep ocean, which again, NASA said it was too hard, and deep space, which we don't really have the technology to physically explore yet, deep time is the final frontier. Do you guys know what I mean by deep time? Yeah, geologic time, so millions and millions of years. Our planet, do we know how old our planet is? Anybody? 4.5. Yeah, 4.56 billion years old. So there's a lot of time there. Billion is more than any of us can wrap our brains around, and we've got 4.5 of them. So deep time is the final frontier. I think that is the greatest mystery we have left on our planet, besides the ocean. That can remain a mystery, I think. So, deep time. Paleontology is the key to figuring out the mysteries of deep time, right? Can we all agree on that? Fossil record, the rocks, they can reveal what these past ecosystems look like and help us understand our place on Earth today and how our planet has changed up to the point where we are now. So, I'm going to take a water break because we're about to get into the real section of this presentation. Is the volume up on your computer, by the way?
I think so.
Okay, cool. All right. So, you guys thought the presentation already started, but joke's on you. The presentation starts, well, now. Welcome to Bones, Stones, and Unknowns, a deep time voyage exploring strange old worlds with your captain, me, and you all as my crew. Time, the final frontier. These are the voyages of the NPS Enterprise. Its continuing mission? To explore strange, old worlds. To seek out extinct life and forgotten ecosystems. To boldly go where no one has gone in millions of years. So what do you guys think? Are you down for an adventure tonight? All right, okay. You're down to be the crew of the NPS Enterprise? Very good. Because I think once the song ends, we'll be at our destination. All right. That was a little bit louder than I was expecting. Could you guys hear what I was saying? Cool. Okay, great. That works out. Okay, so it seems as we're approaching our destination. Shall we begin? Are you guys ready? Okay, let's beam down and check out where we are so we can learn what it's like to do paleontology, to be a Park Service paleontologist. We'll beam down to a mysterious planet called Earth. Okay, Captain's log, stardate, 32.54 MYA. My crew and I landed on Earth, but it wasn't the Earth we knew. This Earth was full of strange creatures, the likes of which we'd never seen. We were supposedly in North America, but it didn't look familiar at all. We came across large cat-like creatures with big saber teeth, horses that were only about 3 feet tall, a tortoise that looked, well, like a tortoise. And then we saw the most amazing thing I've ever seen. The teeny tiniest, most adorable deer you can imagine. They're like 5 pounds, you guys. They're tiny. They're so cute. And as my crew and I were looking at these deer, we decided we absolutely have to know more about them. Right, guys? Right? Okay, cool. So my crew and I, we approached with caution to try to learn more about this incredible species we've just fallen in love with. And then something horrible happened. As we were walking up, out of nowhere, one of them keeled over and died. Right in front of us. And obviously, we all screamed and gasped. in shock, which scared the rest of the herd away. And we tried to go find them so we could learn more about them, but they were nowhere to be found. We'll keep looking. End of captain's log. Well, guys, we tried. We tried to find them, but they've run away. I don't know about y'all, but I'm pretty determined to learn more about these little creatures. How about you guys? Yeah? Okay. I'm pretty invested. What did you say? Pick up the dead one. Well, we're not biologists, so there's really only one thing we can do here with our set of skills, and that's wait for it to fossilize and come back later. So, let's talk about being a fossil. What does it take to become a fossil? Anybody know the first step? Dying. Our little deer that we saw is already halfway through the first step. First step to becoming a fossil is you gotta die. Second step is, or part 2 of step 1, is decay, some decay. Too much decay and then you lose your entire fossil, right? Most things never become a fossil. Usually things decompose away entirely and they never end up in the fossil record. So if we want our tiny deer to remain in the fossil record, it's gotta decay a little bit, but not so much that the bones kind of end up eroding away or being washed away. Anybody have any ideas what step 2 might be to the process? Mud? Yeah, something like that. It's got to get buried somehow. And then what happens after that? This is the hard part. Calcification. I like that answer. Yeah. It may not be calcium, but if it stays buried long enough, the groundwater percolating through the ground carries with it tiny minerals in solution. And those minerals pass through the soil, they pass through the rock, and eventually they'll pass through these bones as well. The water passes through, the minerals percolate out and kind of replace the bone as they go. They replace that organic material. This is a process called permineralization. It's essentially turning the bones to stone. So really, when you have a fossil, it's not any organic material that remains. It's a perfect replica of what was once there. So once something gets entirely fossilized, entirely replaced by minerals, what has to happen as the last step for us to find it? Word for word, exposure. Nice. Exactly. So the sediment has to erode away so that we can see it on the surface so that we know that it's there. Otherwise, we'd have no idea. But we don't want to hit step 4 too early, because if you hit step 4 before it's completely fossilized, then the fossilization process stops. So there's a lot that can go wrong here, but we're hedging our bets and we're hoping that our tiny deer can make it through this process that might take, well, it might take a long time. You guys thought you were here for like an hour presentation? We're gonna be here for like a few million years. Okay, so let's see, let's put all of this in a little bit of context. So I wanted to give you guys a look at this time scale. This is the Cenozoic era, which is the era we are in currently. And the Cenozoic started kind of with a bang about 66 million years ago. Something rather dramatic happened. I don't know if you guys heard about it. It was kind of big news. A big old asteroid hit the Earth and wiped out all of the non-avian dinosaurs. And then the Cenozoic began, because there was a huge, huge overturn of the animals that occupied the Earth, right? A lot of life was wiped out then. It was a mass extinction, but we recovered, which is why, I might add, mammals are cooler than dinosaurs. We're still here. I know when you guys heard paleontology as the topic of conversation tonight, you might have been excited that I was going to talk about dinosaurs. I hate to disappoint, but I don't study dinosaurs. Okay, so the Cenozoic is 66 million years long. We talked about Earth is 4.5 billion years old. So let's think about that for a moment. If you take all of Earth history, all 4.5 billion years, and you scale it down to a 24-hour day, so it's more, digestible for our brains, and Earth forms at midnight on the dot. The first single-celled life doesn't even evolve until about 4 a.m. Photosynthesis shows up at like 5:30. Our tiny deer doesn't show up on that time scale until 11:45 p.m. So, our tiny deer falls right in the middle here, tiny deer time. We were around 32 million years ago. 32 million sounds like a lot to us, but it's like the last 15 minutes of Earth's history on that 24-hour scale. So it's still pretty recent. So we're exploring deep time, but not that deep yet. We're just dipping our toes in, right? And for context, do you guys see this tiny white line over here? That's us. And all of written human history. All in that tiny white line. So, time is big, you guys. Time is very long. And with any luck, our deer will fossilize, but we might have to hang out for a while. So, I think I'm going to call up to the Enterprise and see if Mr. Spock can do some calculations for us to see how long this might take. Mr. Spock?
Yes, Captain.
Any idea how long the fossilization process might take on our tiny deer here?
Well, according to current rates of sedimentation, fossilization, and erosion, it should take about 32 million years. If it fossilizes at all, that would be highly illogical, as fossilization is the exception to the rule, not the rule.
Yes, that's very true. Thank you, Mr. Spock. We did cover that. Okay, well, I don't know about you guys, but I don't have 32 million years to wait around. So, I've got a crazy idea, if you guys are willing to hear me out. Are you guys still on board, or have I lost you all? No? You're on board? Okay. All right, well, let's head back to the ship. So what I'm thinking is, what if we fly really, really fast and slingshot around the sun, and then we'll travel back in time or forward in time or something like that, right? I'm really more of a Star Wars fan. I don't know why I chose this as my theme. But what do you say? Should we try it? I think it'll work. Okay. Let's see. Okay, I'm gonna everybody hold your breath, because I don't know how this is gonna turn out. We might fly directly into the sun. We'll see. Okay. I think it worked. All right. We should be good. Okay. So now we'll try landing on Earth again, but in the future, which for us is still the past. Ah, very nice. Captain's log. Stardate, 2016 CE. We appear to have landed just where we left our tiny deer, but 32 million years later. My crew and I, we plan to explore this area and look for the fossil remains of the tiny deer we all immediately fell in love with. This land is now federally operated. This is a place called Badlands National Park. So, we're lucky because Badlands National Park allows visitors to walk around wherever and look for fossils, as long as we properly report what we find. So, with any luck, we'll find something and have it to report. Okay. All right, you guys see anything? We're looking for a skull about yay big. It's kind of beige, dark brown teeth. Anybody seen it? Off to the left. I don't see it over there. Maybe we can phone in some help again. Mr. Spock?
Yes, Captain.
Can you do a scan for us?
Okay, we've scanned the planet's surface.
Any sign? Any sign?
It appears the fossil is up on the hill to your right.
Up on the hill to the right, okay. Oh, there it is. Right there. Perfect. Oh, just as cute as ever. Well, since we found our fossil, but since we are in a national park, there's a certain set of steps we have to follow. Does anybody know what to do if you find a fossil in a national park? There are 3 steps. Anybody want to take a guess at the first one? You don't get to answer. Because if you answer, you might get a tiny dinosaur. Just saying. First step.
Don't touch it.
Don't touch it. Leave it exactly where it is. Would you like dinosaur? Nice.
Thank you.
Well, very nice. Step number 1 is leave it exactly where it is. Because paleontologists need to know what layer of rock it's from to be able to tell how old the fossil is. We don't date the fossils, we date the rocks around them. But also, another reason to leave it where it is you don't know exactly how to excavate a fossil, right? There's very certain procedures, I don't know, maybe some of you do. There are very certain procedures that have to be followed, and you risk damaging the fossil if you do it incorrectly. You also risk potentially leaving some of it behind that you didn't see. So, very important to leave it right where it is. Any ideas on step number 2? One step before that.
Use your GPS or something to locate where it is.
GPS, yeah, that's half of step number 2. I think you said it earlier. Photos, yeah. Bingo. So you take photos. I always tell people if you want to get an A+ in taking photos, you get something for scale in your photos. Any common object that people know the size of, like a coin or a pen, something like that. Not your hand. We don't know how big your hand is. And then you also get photos of the surrounding landscape too. That's very helpful. And then, yes, exactly as you said, GPS coordinates. Would you like a dinosaur?
Maybe give the person with the photograph one to.
Awesome. So we get the photos, we get the GPS coordinates, we left it exactly where it was. And then step number 3, who said it? You said it. You already said it. Awesome. So step number 3 is, can you repeat it for us?
Report it.
Report it. Bingo. That one matches your jacket. Oh, perfect. That's dino fashion. Oh, perfect. So report it to the rangers. Usually at national parks or really any public land, you can go find a ranger in person, usually at a visitor center, and let them know what you found. Some places, like really cool national parks, like Badlands National Park, give you neat stickers that say, I did the right thing, and have a saber cat skull on them. Just saying. It's pretty cool. And you can also sometimes email your finds in if you can't make it to a visitor center. So that is exactly what we're going to do here. So we'll let the rangers know. We did all of these steps, and we'll let them come out and dig it up. Sound good? Okay, very important. Now we've contributed to citizen science. Very exciting stuff. All right, so in the meantime, because I don't know if you guys know this, but paleontology is kind of a long process. It takes a long time. So we've got to wait for the rangers and paleontologists to actually go out there, and then we got to wait for them to dig it up, and then we got to wait for them to take it back to the fossil preparation lab, which is where paleontologists then use these teeny tiny tools to very meticulously remove all of the dirt and rock and sediment from around the bones to expose as much of it as possible so that we can see all of the features. So it might be a minute, but it won't be 32 million years, which is why we did it this way. So in the meantime, since we're pretty excited about our tiny little deer skull, I think we could do some research. What do you guys think? Learn a little bit more, get some context, just in case it's scientifically significant, then we'll be ready to go, you know? Okay, so first thing, let's talk about evolutionary relationships and how we classify life. Have you guys heard of you guys remember the whole taxonomic rank thing? Maybe you learned this in school: kingdom, phylum, class, order, family, genus, species. Does anybody have a fun memory device for this? You have one?
King Philip came over for green spaghetti.
There you go. I can never remember them, so which is funny. I just remember the original thing. So why is this helpful? We like to, as humans, put things in little boxes because it helps us understand them, and it helps us categorize them and to understand who's related to who. So we have all of these different levels of classification, and as you go down the row here, they get more specific. The most specific category being species. That's why it's called species, because it's the most specific. So that helps us figure out how things are classified, who they might be related to, and what we call them, their naming schemes. That's helpful to know too. Let's try an example out here. I talked to the Badlands Rangers, and they told me that this animal right here is the most common fossil found at Badlands. Does anybody besides Nikki or Max know what this animal is? I would be super impressed. He got it. You work for the Park Service, though. That's kind of cheating. But I'll give you a dinosaur, which is ironic because this is, again, not a dinosaur. So that is an oreodont, which is a very, very bizarre animal. At Badlands, we like to call them our deer, sheep, camel, pigs, because they remind us a little bit of everything. And though we don't know exactly who their closest living descendants are today, we do know how to classify them still. So let's practice with our oreodont here. Kingdom, Animalia. Pretty self-explanatory. It is an animal. Phylum, Chordata. That's a word we might not all be familiar with. Basically, they've got a backbone, right? And some other fun stuff going on. Class, Mammalia. It's a mammal. What makes a mammal a mammal? Live birth. What did you say? Hair. Warm-blooded. Yeah, classics, just like us. Order, Artiodactyla. Any idea what that might mean? Toad, yeah, dactyl, yeah, exactly. So artiodactyls are even-toed hoofed mammals. So they've got either 4 or 2 toes on each foot. Yes, hoofed. Yeah, that only applies to the hoofed animals, or hoofed mammals, I should say. The family here is called Mariquitidontidae, which is a mouthful. And the genus is Mariquitidon. So, that genus contains a couple of different species. If we took a look at this skull in real life, and we looked at all these tiny little features, we would be able to distinguish which species it was. And in this case, it's Mariquitidon culbertsoni. So when you refer to the scientific name of an animal, whether it's alive or whether it's extinct, you refer to it by the genus and species name. So we would call this guy Mariquitidon culbertsoni, which again is a mouthful. So oftentimes you'll hear us abbreviate it as M. culbertsoni. Okay, very good. Any questions about that? We've kind of done a lot. Yeah, you got one?
Culbertsoni, is that somebody's name?
Yes, yeah, good question. So it's named after this guy, Culbertson, yeah. A lot of times, especially in paleo, a lot of stuff gets named after people or places, either the place where they were found or somebody significant to the discovery in some way, but not the author of the paper. You're not allowed to name it after yourself. That's against the rules. Very frowned upon. The other thing they would get named after would be a feature of the animal. I'll have an example for you on the next slide. So we got that. Let's talk about our tiny deer, because surely in the last 32 million years, somebody has researched these things, so we can learn a little bit about them by going out there and seeing if there are any papers. And I'm here to tell you that our tiny deer are part of the family known as the Leptomoricidae. And lucky for us, but perhaps unlucky for science, I only found like 8 or 9 papers ever about the leptomoricids. So we can become experts really fast. But there's not a whole lot of info out there. These things have kind of been understudied, as a lot of fossil mammals are. So, Leptomoricidae. I went ahead and read all of those papers for you guys, so you didn't have to worry about it tonight, but I would love my crew to read them at some point if you get a chance, so I'll send them to you. Okay, but the Leptomoricidae, they're mammals, and again, artiodactyls, so they're part of that even-toed group. And the Leptomoricidae themselves are a family of small, hornless, two-toed, deer-like ruminants. So I've been calling them a deer, but they're technically not deer. They're deer-like. But I'm going to keep calling them a deer because it's easier. Sound good?
Sounds good.
Cool. Okay, and then within the Leptomoricidae, there's a couple different genera. The first one I'm going to talk to you about is called Pseudoparablastomyrix, which, wow, I don't know what they were thinking when they came up with that name. That is a lot. It is maybe the biggest mouthful. I have had to write that so much that my phone and my computer now know that spelling for autocorrect. Yeah, so really fun. But again, we talked about we can abbreviate that when it comes to the species name. Pseudoparablastomyrix, that genus only contains one species. That is Pseudoparablastomyrix scotti, or as we'd like to say here, P. scotti. And then we've got Leptomyrix. That's another genus within the Leptomoricidae. And Leptomyrix has a bunch of species, actually. I just put 2 up here to give you some examples. Leptomyrix evansi, which is the most common leptomoricid found at Badlands National Park, and Leptomyrix ellisei. Now, in general, the leptomoricids, they ranged from like Saskatchewan all the way down through Mexico, the entire Great Plains back in the day. By back in the day, of course, I mean 46 to 13 million years ago. So they were pretty prolific, and to this day, they are one of the most common fossil finds at Badlands National Park. That being said, what is usually found is just their jaws, just their teeth, their lower teeth specifically. It is exceedingly common to find those, and yet exceedingly rare to find a skull, which we just did. Yeah. So nice. So it might be significant. We'll see. Yeah, as we've been reading up on these things, we're learning that our find might actually contribute to science. So, what else? Oh, but wait, there's more. There are tiny deer alive today. There's this fun group called the tragulids, which are also just, wow, so adorable, right? They're the chevrotains, the mouse deer. Has anybody heard of these before? They're so cute. If you ever see one in person in like a zoo, or if you're ever in Southeast Asia, I guess, they're amazing. They look like little potatoes with toothpick legs, and I love them. So they're part of the tragulid family, which is the smallest hoofed mammals in the world. And there are 10 species that exist, divided among 3 genera. Mostly they occupy South and Southeast Asia, but there is also one species in Western Africa, the water chevrotain. So it's pretty interesting. We've got our fossil tiny deer in North America in the family Leptomyridae, and we've got our modern tiny deer over in mostly Asia in the tragulid family. Tragulid fossils have never been found in North America. Never. Only leptomyricids. So keep that in mind. It might come back later. All right. Okay, we've got a good set of background knowledge. Anybody have any lingering questions about tiny deer? I know that was a lot of info all at once. Okay, feeling pretty good. You have one?
Is there any particular reason that we think that it's only the lower jaw that's found and nothing else? Is it harder? Any ideas on that?
Great question. So when things preserve in the fossil record, it can be a little bit weird. Sometimes there's these weird preservation biases almost. And a lot of the times at Badlands, for example, we find a lot of skulls by themselves with no postcrania, none of the rest of the skeleton. And that's probably because that skeleton got washed away or eaten, whatever. The skull becomes the heaviest part, so it sticks around, which is why it's so weird that the leptomyricids, we don't find their skulls. You'd think that that would be the thing that would stick around, as we're seeing in a lot of other animals. Because in mammals, a lot of times you don't find the skull and the jaw together because our jaws are only connected with muscle, right? They're prone to falling out if you're decomposing. So for whatever reason, the jaws that got separated from the skulls are preserving and the skulls themselves aren't, which is fun. Paleontology is full of mysteries. There's still a lot left to solve and figure out. You have a better answer than that? Cool, okay, just making sure. It's also a group activity. Science is a collaborative thing. We all work together, so. Very good question though, because that's one that like literally keeps me up at night. I think about that a lot, so. Okay, the other very important thing that we, as the good scientists we are, must include in our field notes and in any research we do is the location where we found our specimen. If you have a specimen without locality data, it's basically useless to science. And this happens a lot. People donate specimens to museums, which is awesome. That's really cool. But if we don't know where it came from, we can't really use it for science. So if you ever end up with something you're going to donate to a museum, please try to figure out exactly where it came from before you do so. It would be super helpful. Now, we've talked about that our tiny deer came from Badlands National Park, which, by the way, if you're not familiar, is in the southwest corner of South Dakota. It's that white blob down there. We can be seen from space, so we're kind of a big deal. And if you zoom in, this is what the park map looks like, the official National Park Service map. And our tiny deer came from this area right here. Now, if this were our actual field notes, we would write down the exact coordinates and the exact area where it came from. But that's not something you share, right? That's something we kind of only share with other researchers or people who are, in the Park Service doing this sort of work. You don't really publish the exact coordinates. Any ideas why? Yeah. Charlie with the hammer, exactly. You might come out and try to find another one. Yeah, fossil poaching is a real thing. So we'll give you the general area, but we won't tell you exactly where. But for our purposes, this is plenty enough detail. But we can't just know where it's from. We also have to know when it's from, right? Because there's a lot of layers of rock at Badlands National Park, and each layer represents a different time period in geologic time, and a different paleo environment. Badlands actually has rock that is 75 to 69 million years old. That's our oldest layer of rock. That is from a time period called the Cretaceous. And as you can see, pretty different environment than what we're looking at with our tiny deer. Our little deer would probably not last very long in this environment. I think he'd get munched up by that guy pretty fast. By the way, if you want to learn more about those giant marine reptiles there, come to tomorrow night's talk, because that's what it's all about. So I'm pretty sure we can count that one out. Up next in our Shadron Formation, that is in the Eocene period from about 37 to 34 million years ago. That paleo environment is kind of like Florida today. It's very swampy, very humid. There were alligators in South Dakota at this point in time, so that gives you a pretty good idea of what was going on. And then up on top in our Brule Formation, we are up in the Oligocene from about 34 to about 28 million years ago. And that, of course, is where our tiny deer came from. We kind of cheated because we already went back in time and we saw that, so we knew what to expect. But if you didn't have the capability to go back and visit in person, you got to figure it out from the rocks and the fossils. So we've pieced together what that environment looked like, and we know that that layer is where our tiny deer came from. But guess what? We have to be even more specific than that in our notes, because we can't just say the Brule Formation. We've got to say exactly which layer in the Brule Formation. And lucky for us, the geologists have already done the hard work, and they've divided the Brule Formation into a couple members, and then they've divided those members into even smaller units. So we're in the Brule Formation, but we're in the pole slide member, and we're in Unit 8 of the pole slide member, which is also called the Bjork Siltstone or the Leptocania beds, which lines up with the earliest Whitney and North American land mammal age, which is in the early Oligocene, and none of that is important for you to know. The only thing that's important for you to know is that that's about 32 million years ago. And it's that layer right there. This, by the way, is the view out of the visitor center, which is pretty cool. So, 32 million years ago. If we take a look at all of the tiny deer that exist around 32 million years ago, to try to narrow down which one ours might be, unfortunately, we're left with quite a few options. These are the ones that kind of exist in and around that time period. There are a lot of different species here. And these little drawings off to the side, hold on, let me get my laser pointer. These little drawings off to the side are their teeth. Because, as we talked about, the jaws are usually the thing that are found. So all of the characteristics that define the different species of these tiny deer are based on their lower teeth. That's it. So, we've got a lot of options here. We've done our research, we've narrowed things down, we've figured out when and where we were, so I feel like we're in a pretty good spot. I don't know, do you guys think we've waited long enough? Should we check in and see if they've prepped the specimen yet? Okay.
Yeah.
Hello, Mr. Spock, are you there?
Hello, Captain.
Any chance they're done with the specimen yet?
They are. They have tentatively identified it as Leptomyrix ellisei.
Oh.
Scientifically significant because it's the first skull.
You don't say. Thank you. Very nice. Okay. Well, great. We've got a new discovery. There is our guy. Looking pretty good. All cleaned up. So, yeah. First skull ever of Leptomyrix ellisei. That's pretty exciting. Good job, you guys.
Yeah.
Yeah, give yourselves a pat on the back. So, all cleaned up and nice. We've got the jaws with the skull. As we mentioned, that is very exciting news because that's hardly ever the case and because all of the species defining characters are based on the lower teeth. So now we can take a very, very detailed look at those teeth to see what kind of characters we have. They've tentatively identified it as Leptomyrix ellisei. We'll see if that's true. I feel like we should research it. What do you guys think?
Research it.
Yeah, it's the first skull ever. I feel like this will be a pretty easy mission. You know, we can write it up, describe it really well, get it out in the written record for other scientists to reference later on. Shouldn't be too difficult, right? Yeah, seems pretty straightforward. It wasn't straightforward. Okay. All right, so to understand which species we're looking at, we gotta take a look at teeny, teeny, tiny characteristics in the teeth. And you guys don't have to remember any of these words, so don't worry. But no test at the end, as far as you know. But keep in mind, the skull itself is only about this big, so the teeth are each this big at most. They are tiny. So we are looking at teeny, teeny, tiny characteristics to distinguish these things from each other. A really common one is this thing called the entoconulid, which is on the end of the wisdom tooth. Whether it's rounded and pronounced or, kind of reduced, that's an important characteristic apparently. There's enamel that can be kind of wrinkled and crenulated versus smooth. There's this little thing jutting out right here called the Paleontology fold. That one's really important too. So there's all these tiny characteristics that help us figure out who's who, and basically you're just gonna have to trust me. Cool? Sweet, all right, because I read all those papers and now it's burned into my memory. So now we get to the portion, everybody's favorite portion of any talk, right? Which is the comparative anatomy portion. Obviously everyone's favorite. Because we've taken a look at our tiny deer and we've taken a look at a couple other specimens now, and things maybe aren't as clear cut as we thought. So what we're gonna do is we're gonna compare our specimen to two other specimens that also have skull material. Because as we talked about, the skulls are exceedingly rare. But we do have a skull of Leptomyrix evansi and a skull of Pseudoparablastomyrix scotti. So we can compare all the characters and see which one ours lines up with best. And to do this, we're going to divide you into two teams. Who wants to be Team Leptomyrix? Which side?
We'll be it over here.
Team Leptomyrix. Okay. You guys are the blue shirts, I guess. Team Pseudoparablastomyrix scotti. P. Scotti. You guys are the red shirts, obviously, Scotty. Okay, so we'll keep score. L, P. Okay, are you guys ready? We're going to run through this pretty fast because it's not the most exciting part of the presentation. So, that Paleomyrix fold I mentioned, Leptomyrix ellisei and Pseudoparablastomyrix scotti have it, but evansi does not. Sorry, guys. Crenulated enamel, again, scotti matches up with our guy. Evansi does not, so that's 2 points over here for the lower molars, but that pesky little interconulid thing I mentioned, that lines up with evansi and not scotti. And since I've done my reading, I happen to know that that characteristic is considered to be pretty indicative of evolutionary relationships, so that one is kind of worth 2 points.
Yes.
Nice. So with the lower molars, we're going to give that a solid tie. Do we feel like that's fair? Point for each.
Whatever.
Up next, we've got the upper teeth. Let's see. Whoa, I did it again. There we go. So Leptomyrix ellisei has a similar structure of the third premolar to Leptomyrix evansi, whereas in P. scotti, the margin of the premolar is kind of expanded. It creates a little bit of a basin, so it's pretty different, whereas the rest of the teeth look pretty similar. So we're going to have to give that point to...
Yes.
There we go, Leptomyrix. Okay, 2-1. Okay, crania. How we feeling, P. Scotti team? You guys feel like you're making a comeback? You ready? Not gonna make it? Okay, so the skulls. We've got this thing called the sagittal crest in the middle of the skull. In evansi, that sagittal crest is really long, but in ellisei and scotti, it is much more reduced. It is way shorter. Kind of similarly to that, the temporal crests, which are the arch part coming off of the sagittal crest, are a lot wider, whereas in evansi, they are much narrower and shorter. I heard a yes. All right. And then the length of the skull. The skulls, each of them are kind of broken off in the front at a different point. So what we did was we measured from the back of the eye socket to the back of the skull to kind of retain consistency throughout the specimens. And so, again, evansi is a lot longer than ellisei and scotti. So I think that was a resounding point for P. Scotti. Sorry, Leptomyrix team. We are back to a solid tie. Now here's the plot twist. We got to introduce another contestant. Remember those modern tiny deer? The tragulids that have never been found in North America? Well, guess what? Our weird little skull that we found happens to have characteristics that have never been seen in the leptomyricids before. It has a couple characteristics that are only present in the tragulids. For example, this little connection point here. See how in evansi it's like half blue, half red? Same down here in scotti. Well, in ellisei, it's mostly the red part, much less the blue. And that is something that has only ever been seen in the tragulids. So that is very weird. So our guy doesn't really line up with either of them. We'll give a point to the modern tragulids, weirdly enough.
I said, who invited him?
Who invited him? That's what I was asking myself at this point in the research project. Yeah, so there's a couple things that could be going on here. Either we're figuring out that maybe the leptomyricids of North America eventually made their way over to Asia and maybe they evolved into the tragulids, but I would never say that based on such limited data. What I would much sooner claim is that this is something called convergent evolution, which is when two totally different unrelated animals occupy similar niches, similar jobs in an ecosystem, they can end up having similar traits because they're doing the same thing, right? So, we can't claim anything too extreme yet, but there's one more characteristic we want to consider here, and that's in the base of crania, the base of the skull. So there's this part here that is very rectangular on Evan's eye. Pseudoparablastomeric scotti is not considered in this one because the base of the skull is wrecked. It's destroyed. Sorry, guys. But these two, that area is much more trapezoidally shaped. So again, another point for the tragulids. So guess where we are again? A tie. This time a three-way tie. So, what do you guys think we should do? Keep going? 5 more? Go back in time? I like that answer. And figure it out. We could just ask him who he is, right? So, before we get to what we're going to do, I know there's one question you've all been asking yourselves and you've all been dying to know. What about pronodens, right? Duh. How do you forget, right? Of course. Yeah, so you can't forget about pronodens. It's such a problem child. Pronodens is another leptomeriscid, and it does fall in between leptomeryx and pseudoparablastomerics in terms of time, which is also where our tiny skull is from, but the teeth are like super worn down. I don't know if you guys can tell, but like this was a very old individual when it died. Super worn down. And you remember all those tiny characteristics we were looking for in the teeth? How are you supposed to see those? They're all leveled out, exactly, gone. So they have no characteristics in their teeth. The only distinguishing characteristics about pronodens are in the front portion of the jaw, which, guess what, is the portion that always breaks off. And none of the other specimens have it. So, as far as I'm concerned, pronodens is out. I don't think this was even valid to name it in the first place, to be honest. No hate on the on the pronodens authors, but it's been a while. I think they published this in like the 20s, so we're good. Yeah, so basically, you guys don't have to worry about pronodens. I know you were all concerned. I'm happy to put your minds at ease. We can move on from that one. Feel good? Okay, great. Cool. So, our conundrum remains. What do we do with our tiny deer skull? What is going on down there? Sorry. As I mentioned, ADHD. Very distractible. Very distractible. Okay, so we've got our tiny skull. We've got characteristics from all 3 of these groups, leptomeryx, pseudoparablastomerics, and the tragulids. Does anybody want to make a case for putting our skull in any one of these 3 groups? Anybody want to make an argument? No? Good. That's exactly what I did. So. Well, they were in such different, the tragulids at least are in such a different time period. And actually, I didn't really mention this, but pseudoparablastomerics is about 10 million years after leptomeryx. Yeah, so time removed us. Yeah. So, no arguments to be made because it doesn't fit into any group. So what we have to do as a research team here, obviously, is put it in a new group. What do we think? Does that sound fair?
Sounds good.
Yeah. It doesn't fit in any other group, we got to create a new group. Here we were thinking this would be a nice and easy mission, when all of a sudden it got super complicated and we got to create a whole new thing. But, you know, worth it. So, we're going to name this group Santusi-Merix. And I will tell you why. Captain's log. My crew and I have just named a new genus of tiny, adorable Oligocene deer. And we named it Santusi-Merix after the head of the Geologic Resources Division for the Park Service, who is a great friend and colleague of ours. Obviously, we all know him very well. And we published a paper. We went through the peer review process all together, and it was a whole thing. Took a long time, but we did it because we were committed, and it didn't take 32 million years. So, we did it. If you guys want to read your fabulous work, feel free to scan here. And we were very happy to see that the general public was a big fan of our tiny deer. And Vince Santusi, who we named it after, he was so excited about it, he even commissioned some absolutely adorable paleo art. Oh my God. Look at how cute. And people were so excited about this tiny little guy that he even got put into the coloring book that the National Park Service publishes. So if you ever see one, grab a copy because you can color your own tiny deer, which is very fun. Okay, I would call that a resounding mission success, you guys. Thank you very much for joining me. Let's do a little reflecting. You guys, I know I've gone way too long, sorry. Yeah, way too long, sorry. We'll do a little reflecting real fast. We found and researched the first ever Leptomeryx LSA skull. That skull displayed traits from multiple genera. We had to assign it a new group because we couldn't fit it into any existing one. And then we named that new intermediate genus Santusi-Meryx. And we, as good scientists, know that in the future other researchers may invalidate our genus. And that's great, because that's how science works. Somebody may come along in the future, and maybe they'll take this absolute mess of a diagram here that has all of these question marks because we've never seen skulls of any of these tiny deer, and they'll find one, and they'll figure out, hey, maybe that guy looks a lot like Santusi-Meryx. Maybe it needs to go in that group. Or maybe we need to switch things around entirely. Who knows? All I know is that before we do any of that, we need more skulls. So, when new information is gained, scientific literature is updated, as we all collectively work towards a more comprehensive understanding of deep time, which is, after all, the final frontier. So our continuing mission as, oh, I forgot that happened. As, so loud, how do we turn this down? There we go. Our continuing mission as the crew of the NPS Enterprise is to do citizen science. Okay, apparently the music cut out. That's fine. Do citizen science. That means when you're out in national parks, report fossils you find. Follow all those steps we talked about. You can get involved in other citizen science projects if you do a simple Google online. There's tons of stuff that you can help out with every day and get other people involved in too. The second thing we got to do is protect public lands, because if we don't, then where are we going to do our science? Protect public lands, and that means more than just the Park Service, the Forest Service, the Bureau of Land Management, Fish and Wildlife, state parks, any public land should be protected and preserved for future generations. For views like this. Again, space is cool, we'll give it to them. For the endangered species that might live on those public lands. I might have stolen the Utah prairie dog photo from Nikki without telling her. And this is the black-footed ferret in South Dakota. Very, very endangered animal there. So, protecting public lands. Our third part of our mission is science communication, which, as I mentioned, I do a lot of for the Park Service. Max and I do a lot of it on our own time, and you guys can do it too. Teach people what you've learned. Doesn't have to be very detailed stuff. You can talk about it anytime and encourage people to get out and do citizen science too. That's part of science communication. Another part of science communication is events like these. There's tons of cool stuff that you can go see all the time, especially at the parks. And then shameless self-promotion, social media. Max and I do a lot of social media science communication under the names Geosplore and Notorious Naturalist Max. We mostly post our stuff together. So we try to do really fun, humorous scientific videos. So if you want to join us and kind of, see what adventures we get up to, because we're kind of all over the place, feel free to follow along. And, of course, don't forget tomorrow night's presentation. It's going to be very exciting, I promise you that. Okay, so our continuing mission: do citizen science, protect public lands, and communicate science. What do you guys think? Can you handle it? Sweet. Why is this mission important? You tell me. Any questions? Sorry, didn't mean to flip. What'd you say?
I was just going to say just for
The future, yeah. Preserving for future generations, that's the mission of the Park Service, right? I think we talked about a lot of reasons why it's important, but does anybody have anything they want to add? Understanding the past, which helps us understand the present, and sometimes actually the future. Yeah. Any other thoughts? Okay.